Hello everybody, my name is Daniel and today I will be presenting my work entitled Effects of MIR146B Gene Editing on Anaplastic Thyroid Cancer Utilizing CRISPR-Cas9N.
MicroRNAs are small non-coding RNAs that regulate the expression of a targeted messenger RNA by inhibiting the translation or promoting the adenylation and decay, being key regulators of processes such as differentiation, proliferation, apoptosis, and metabolism. MicroRNAs are commonly deregulated in cancer, and this group of microRNAs is known as oncomers. Oncomers can both be down and up regulated, and both of these deregulations can lead to cancer, as shown in the image. In thyroid cancer, MIR146B is the most overexpressed microRNA in both papillary and anaplastic carcinoma, being highly associated with tumor aggressiveness and recurrence, leading to a worse prognosis. Having said that, our aim is to understand the role of MIR146B in an aggressive thyroid cancer model using CRISPR-Cas9N gene editing methodology. The KTC2 cell line derived from anaplastic thyroid carcinoma, which has a high expression of MIR146B, was selected for this study. The silencing of MIR146B expression was performed using the gene editing methodology CRISPR-Cas9N. The Cas9N nuclease is mutated in the root C domain, becoming a nicase. Thus, for the DNA double strand to break, two single guide RNAs are needed to direct the Cas9N to opposite strands of the DNA, flanking the desired addition site. The guides were cloned into two distinct plasmids, which were co-transfected using lipofectamine. A control group of transfected cells with no guide, known as empty vector, was also established. Having no guide, the Cas enzyme has no target, thus not promoting any DNA cleavage. Our first step after selecting the clones was to measure the expression of MIR146B by RNA quantification. The level of MIR146B5P went down by roughly 40 and 25% on the test normalized by U6 and 60 and 30% on the test normalized by R and U6B compared to the control in clone 1 and 3 respectively. After the confirmation of lower quantities of MIR146B5P in the treated groups, the in vitro tests were performed. This way, cell counting assay would be our first test. This way, 20,000 cells per well were transferred to a 12-well plate in sextuplicate and grown for 24, 48, and 72 hours. The average value of the sextuplicates was determined by counting the cells in a cytometer. The viability of the cells was then measured using the MTT salt. The higher absorbency indicates there are more functioning mitochondria that turn the MTT salt color to blue. Considering that cells were plated in a 96-well plate at a density of 1,000 cells per well. The absorbance reading was then carried out on a spectrophotometer and the average of the octuplicates was determined. Here it is shown that the absorbency of the control group was higher than the treated, indicating that the cells in the control group were more viable. The next assay performed was the wound healing. In this test, cells were cultured in 6 cm plates until they reached confluence, being then treated with mitomycin to block cell proliferation. We then analyzed the time necessary for the cells to fill the gap, and representative images were taken with the aid of a microscope. After 22 hours measuring the wound, the control group cells finally migrated to and closed the gap, but a very noticeable difference can be seen in the treated group, as shown in the images. The difference was also quantified and illustrated in a graph, showing the percentage of area closed by both groups. We then analyzed the capacity of the cells to form colonies. 500 cells were transferred to 6 cm plates and expanded for 8 days. As we can see in the image, formation of colonies was very reduced in the treated group. As many studies have shown, MIR146B is a highly upregulated microRNA in thyroid cancer, contributing to aggressiveness, invasion, recurrence, and overall a bad prognosis. The downregulation of this microRNA in both follicular and anaplastic thyroid cancer has shown a decrease in all the characteristics mentioned above, leading to a better possible prognosis. Thus, this study aimed to show that the gene editing of the MIR146B gene via the CRISPR-Cas9N tool could also reduce the expression of MIR146B and lead to the same results of reduced aggressiveness and all other characteristics related to the disease on a molecular and cellular level. Thus, This study showed that CRISPR-Cas9-N mediated gene editing is efficient to reduce miR146b expression, resulting in reduced migration, cell count, viability, and colony formation, possibly also reducing overall aggressiveness. Thus, molecular targeting of miR146b through the use of the CRISPR-Cas system could be a potential therapeutic approach for aggressive thyroid carcinoma.